She'll log in, maybe she won't. So, share screen. Come on. There we go. All right. Okay. All right, it is time to commence, so we'll go ahead and start. Let's begin this day with a word of prayer. Father in heaven, thank you so much that you have seen fit to redeem us, not with perishable things, not with gold or silver or anything else. You've redeemed us from the ways that might have been inherited by our ancestors, but no, you redeemed us with the precious blood of Christ, a lamb without defect or blemish. We thank you, Lord, that we are yours, that we are chosen through the foreknowledge of God the Father, through the sanctifying work of the Spirit, to be obedient to Jesus Christ and sprinkled with his blood. That grace and peace is with us in abundance. Thank you. Now we pray for those who are ill. Please heal them. And for those of us here today, help us to concentrate and learn. Help me be clear. We thank you for this time in Jesus' name. Amen. Okay. So we looked at the definition of natural and the definition of synthetic, and we saw that your body doesn't care. If it's a natural synthetic poison, it cares what the molecule is. It doesn't care where it came from. It will use vitamin C, whether it's synthetic or whether it came from an orange. Doesn't care. It'll use vitamin D, whether it came from yeast extract or whether it came from a tablet. Your body doesn't care. So whether it's natural or synthetic, your body doesn't care. If the poison came from a natural source like a jellyfish or from a pill, it'll still kill you. It'll kill you the same way, and your body doesn't care. So with that in mind, we're going to see that molecules interact with targets. And those targets are enzymes and receptors and things like that. And interacting with these targets is going to depend a great deal on the shape of the drug, the 3D shape of the drug. Now, one of the things that determines the 3D shape of the drug is its stereochemistry. And stereochemistry is a branch of, in particular, organic chemistry that helps us describe the shape of a molecule in 3D space. So to illustrate this concept of enantiomers, I have these two makeshift molecules here. So if you hold these here like this, you can see they're mirror images of each other. So if we stick a mirror right in the middle here, this is a reflection of this one. Now, we take these two reflections and now we try to superimpose them. Can they superimpose? No, they can't. So they're mirror images of each other and they're not superimposable. We call such molecules enantiomers. Enantiomers. So in terms of their chemical formula, they have exactly the same number of carbons, exactly the same number of hydrogens, exactly the same number of nitrogens, oxygens, whatevers, but their shape in 3D space is different. And if their shape in 3D space is such that they're mirror images of each other, they're enantiomers. If they have the same empirical formula, but they're not mirror images of each other, they're called diastereomers, not mirror images of each other. Now, you're probably wondering, what is he going on about? And what does this have to do with pharmacology? A lot, actually. Because you can have two drugs that are chemically the same in terms of number of various types of atoms. Also, the way those atoms are bound to each other is virtually the same. It's just their 3D configuration is different. And that will have profound implications for which targets with which they interact. So I have here two molecules, and these are actually a molecule. If you like to cook, you probably have had some experience with this molecule. This is tartaric acid. Now, if you've ever used cream of tartar contains bitartrate, these molecules. So if you've made tartar sauce for your fish, or if you had a filet of fish sandwich from McDonald's, or if you've been to McDonald's, it's okay, I forgive you. But the filet of fish will have that special sauce, and it has tartar sauce in it. That kind of bitter taste that the tartar sauce has. Some people put tartar sauce on their eggs. If you do that, I forgive you too. You guys are looking at me funny. You guys have had tartar sauce, right? You know what you don't like it. Okay, but you know what it is. Okay, you're looking at me funny as if to say tartar sauce. Not a fan, but you know what it is. Okay. See, when it comes to fish, I'm kind of a purist. I don't even like lemon on my fish. To me, it's an insult of good tasting fish. And the lemon is put on because some fish have a strong aftertaste, and the lemon is meant to take it away. I like the aftertaste. So to me, to put that on there is to defile good tasting fish. So I'm a bit of a purist when it comes to my fish, but that's just me. Same thing with lamb. To me, lamb is the world's most delicious meat. To put mint flavored jelly on it is a complete crime against meat. That's my opinion about lamb. I absolutely adore lamb. I would eat lamb morning, noon, and night if I could, but I don't. Yes? Yes, definitely. Most definitely. Now, these two molecules of tartaric acid, in fact, are enantiomers. They're mirror images of each other. If we put these two folks across a mirror, they in fact reflect each other. And so they're both tartaric acid, but they are different stereoisomers of tartaric acid. And there is a way to classify different stereoisomers, and here's how you do it. Now, I'm trying to stand here because it's recording the video, so I wanted to see this, because when Brooke watches this on the video, I want her to be able to see this. So you have a stereoisomer if you have a carbon to which 4 different constituents are attached. 4 different constituents are attached. So if we had 2 hydrogens attached, and the black, by the way, is carbon. If we had 2 hydrogens attached to this carbon, would that be a so-called chiral carbon? No, because you have to have 4 different constituents. Now, if we look at this carbon, we have a OH, a hydroxyl group here. We have a hydrogen here. We have a carbon to which you have different moieties attached to another carbon that's part of a so-called carboxylic acid. We have a C, double bond O, bond OH. That's a carboxylic acid. So that's 4 different constituencies. This carbon here also has 4 different constituencies. So you have 2 so-called chiral carbons. And the number of stereoisomers that a molecule can make is equal to the number 2 raised to the number of chiral carbons. So if we look at bitartrate, tartaric acid, there are 2 chiral carbons. Therefore, there are 2 raised to the power of 2. 2 times 2 is 4. So there's 4 potential stereoisomers for tartaric acid. And if you have just 1 chiral carbon, then you have 2 possible stereoisomers. So to classify the type of stereoisomer, you take the chiral carbon and then you assign priorities to each of the constituents attached to it. The priorities will be assigned according to molecular weight. Now, fortunately, we have a periodic table over here that'll tell us molecular weights. So I have a hydrogen, oxygen, and then 2 carbons. Okay, carbon ties, right? Because carbon is molecular weight 12. So how do you break the tie with the carbons? You go to the constituents that are directly attached to the carbon. We have 2 oxygens here. We have an oxygen and a hydrogen and another carbon. Oxygen beats carbon. So this will be the next highest constituency. So it's going to go 1, 2, 3, and then hydrogen is the loser because hydrogen's the lightest of all the elements, 4. 1, 2, 3, 4. Then you take the number 4, the one that loses, you hold that away from you. And then it's 1, 2, 3. It goes like this. Now, you take that direction and you ask yourself, so if I face this towards me and it's 1, 2, 3, does the clock go that way? Tick tock, tick tock, tick tock, tick tock, tick tock, tick tock, tick tock. Look at the clock. Does the second hand go that direction? Yeah. So that's the right way. That's the way the clock goes. That's the right way. So it's R for the right way. Now let's take this one. So here's 1, 2, 3. Hold it away from you. 1, 2, 3. Does the clock go that way? No, that's the wrong way, stupid. So it's S. So it's a stupid way. You have the right way, where the clock goes, and the clocks don't go that way, stupid. So it's S. You have R and you have S. So stereoisomers can be either R or they can be S. If there are, the constituencies can be ordered in the way the clock goes. That's R. If they go opposite the way the clock goes, well, that's stupid because clocks don't go that way. So it's S. The right way, the stupid way. R or S. And so tartaric acid has 2 chiral centers. Tartaric acid can be RS or SR, or it could be RR or SS. That's 4 different stereoisomers. That's exactly the number of stereoisomers we predicted, isn't it? Now, what does this have to do with pharmacology? I'll show you. Here is a blood pressure medicine called carvedilol. The trade name of this drug is Coreg. Coreg. We don't deal with trade names very often. That's why I have to stop and think about the trade name sometimes because I don't remember it sometimes. There are times when I will say when the trade name is, I forgot. Some drugs don't have trade names because they're available in generic, and they've been available in generic, so the trade name, the trade brand is not available anymore. For example, the painkiller, fentanyl, the transdermal patch, used to be called Duraseg. Duraseg's not made anymore. It's just generic dermal fentanyl, or there's the trade name now, Actic, A-C-T-I-Q, Actic, which is kind of clever, I suppose. So that Duracig, even though people call it that, it's not made anymore in the US because it was discontinued. It's generic. So here's carvedilol. And if you look at carvedilol, that carbon right there is a chiral carbon because you have a hydroxyl group, then you have a hydrogen, and you have two carbons. So you need to break the tie between those two carbons. So now I ask you, which of those carbons is going to win? The one with an oxygen attached to it or the one with a nitrogen attached to it? Well, look at the periodic table. Which is heavier, oxygen or nitrogen? Oxygen. So who's going to win? The carbon attached to a oxygen. So that means it's going to go 1 with the hydroxyl group, 2, 3, and then 4. And because the way the carbons are attached to a central carbon, they're going to form a tetrahedral structure like this. So that means this is actually going to look like this in 3D space. So this will be 1, 2, 3, 4. So if you hold that away from you, it's going to be 1, 2, 3. Is that the way the clock goes? No, if you have to take your finger and do that, go ahead. Is that the way the clock goes? Yeah. So that would be the R stereoisomer. That's the right way. Turns out with this drug, one of the stereoisomers has activity, the other doesn't. But the drug that you take is a mixture between the two stereoisomers because stereoisomers are very hard to separate from each other. And so if we ask which one has priority, if we break them up like this, we agree that R1 has priority over R2. And so then we put it into the stereoisomer mix like this, which is R, which is S, and we see that these two are stereoisomers of each other like that. And you could essentially break it down to moieties like this. And if you struggle with this, the best way to do this is to make models of them. We have Tinker Toy kits. I call them Tinker Toy kits. They're called molecular modeling kits. We have the molecular modeling kits upstairs in the conference room. I have one in my office. If you want to come play with my toys, feel free to come play with my toys. In fact, for the exam, I will bring my kit down and you can make molecules and you can use them for the test. Okay? So, because I think being able to do this in your head is useful, but being able to see it, I think for some people is just money. So, first of all, is there anything about this that's unclear? Let me see if, yeah, Bracken? Chiral carbon is a carbon to which you have 4 different chemical substituents attached. Has to be 4 because, so if you have a carbon with a double bond, you can have stereoisomers, but they're very different types of stereoisomers. Then you have what are called cis and trans isomers. Those are very different because the double bonds are planar. This has the carbon because of the way, so carbon has 4 singlet electrons with which it can participate in bonds. So it will form a tetrahedron like this. And those tetrahedrons, depending upon the moieties that are attached at the corners, will have a different 3D shape. If they have the same empirical formula, they can be mirror images of each other. And then therefore they have a different 3D structure. That means they'll bind targets differently. And if we look at carvedilol and we look at their, its affinity for receptors, because this is an inverse affinity, the higher the number, the greater the affinity for the receptor. You can see, look at that difference between the R and the S stereoisomer when it comes to the beta receptor. I mean, it's huge. It's just huge. So even though you take a racemic mixture of this, it's the R1 that does all the work at the beta receptor. Whereas at the alpha receptor, the affinity is pretty similar. How many of you have heard of this drug for acid reflux disease called omeprazole? Prilosec. The drug Nexium is called esomeprazole because omeprazole is a mixture of R and S omeprazole. Nexium is esomeprazole because it's the S stereoisomer that actually does all the work. So the company marketed it, discovered a way to isolate the S isomer, kept that under its hat. When the patent ran out for Prilosec, they released it as Nexium. Kind of a nice way to get the most out of your investment there. So Nexium, that's why Nexium, the dose for Nexium is half that of omeprazole, of Prilosec, because it's just purely one molecule and that molecule has all the activity. Now, take a look at these molecules here. So the molecule on top is called carvone. There's two stereoisomers of carvone. The R stereoisomer of carvone is used to flavor spearmint. So if you think of spearmint gum or any sort of spearmint flavored, really anything, breath lozenges, mouthwash that you might use, Scope, for example, uses that. And now look at the S stereoisomer, which is S-carvone. That is the flavoring element in caraway, which was used to flavor caramel. So think of your caramel sundaes. Think how differently your caramel sundaes taste from Scope. That's due to the difference in orientation around a single carbon. Folks, a single carbon causes that distinct flavor difference and smell difference. And then down here, we have a small peptide. It's a methyl ester of a small peptide. And this is aspartame over here. It is the L, you use L and D for amino acids, the L being the S and the D being the R. So this would be the S stereoisomer. This particular stereoisomer is very sweet. Sweetness per unit weight, it's about 1,000 times sweeter than sugar. So you use very, 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 very small amounts of it to sweeten materials, to sweeten foods. If you change a configuration around one carbon, just one carbon, that carbon right up there. Now this stuff is as bitter as the day is long. It's so bitter, you put a little bit on your tongue, it's just really bitter. Configuration around one carbon will change that from really sweet to really bitter. Have I convinced you that stereoisomers are important for pharmacology? If that doesn't do it, look at this drug, thalidomide. You guys are way too young to remember the thalidomide disaster. So this drug was outlawed in the United States. It was a morning sickness drug. It was outlawed in the United States because animal studies suggested that it would cause birth defects. But the drug was marketed in Europe, and you started having children born without their limbs. They lost their limbs. Have any of you heard of a guitar player named Tony Melendez? Tony Melendez was a thalidomide baby. He has no arms. He plays guitar with his feet. When Pope John Paul did his United States tour, Tony Melendez played for him. And he plays guitar with his, he plays very well, in fact, but he plays with his feet. He has no arms because of thalidomide. And the morning sickness relieving capacity of the drug comes from the R stereoisomer. The teratogenic part comes from the S stereoisomer. Had they been able to isolate the one stereoisomer over the other, the drug wouldn't have been a problem. It was because the drug was given as a mixture. By the way, mixtures of R and S stereoisomers are called racemic mixtures. Racemic mixtures. Because the drug was given as a racemic mixture, it caused those defects when given to pregnant women. So this will be important for some of the things that we will look at. Let's talk a bit about drug movement through the body. The ability of the drug to be distributed through the tissues, because blood is an aqueous solution, will depend upon the solubility of the drug in the blood. Some drugs get around this by binding to serum proteins or being carried by certain types of particles in the blood that are known to carry lipid-soluble molecules anyway. For example, steroid hormones, estrogen, testosterone, things like that, they're carried by a steroid hormone carrier. It's called the steroid hormone globulin. Steroid drugs also get carried by that steroid hormone globulin. The ability of drugs to penetrate into tissues is going to depend upon barriers, and their ability to penetrate that barrier will depend greatly on the thickness of the barrier. So if you think about drugs getting into the central nervous system, they have to get through the blood-brain barrier. They're very specific receptors the drug may have to bind. And so there are different tricks you have to use to get the drug through the blood-brain barrier. And some of the tricks that people use are quite ingenious. Now, pH is going to greatly influence the ability of drugs to permeate the body. pH will often determine whether something can penetrate certain compartments as opposed to others. pH will also determine how the drug is excreted at times. So many of the drugs we will look at will either be weak acids, a lot more of them are going to be weak bases. And the tendency for a drug to be ionized can be predicted using this handy-dandy equation called the Henderson-Hasselbalch equation. And as you look at this equation, you probably say, I have no idea what that means, but very simply, it goes like this. So this number here, the pKa. The pKa is the pH at which the concentration of the version of the drug that's charged equals the concentration of the drug that's not charged. So the amount of drug you have in the body, that concentration that you have, either in the body or at that particular compartment, the pH at which the charged and uncharged moiety, those concentrations equal each other, that pH is the pKa of that drug. If the pKa is really low, that means your drug is an acid. If the pKa is really high, that means your drug is a base. If the pKa is just sort of low, then it's a weak acid. If it's just sort of kind of high, then it's a weak base. And if it's around about 7, it's neither, it's neutral. Now, just dawned on me, I made an assumption. Do you guys understand chemically what an acid is? Is there anybody who needs a refresher on what an acid is? Everyone understands what an acid is? Yes? All right, so We're going to use the Bronsted definition of an acid, not the Lewis definition of an acid. So a Bronsted acid donates hydrogen ions. Notice it donates the hydrogen ion. That's what we mean by a Bronsted acid. When we're talking about drugs being acids, it's the Bronsted definition of acids. Bases, therefore, will be drugs that have a tendency to take up hydrogen ions. That will be a base. So acids will donate hydrogen ions, bases will take up hydrogen ions. Now, when you're developing a drug, let's say Bracken here is COO of Merck, and someone brings him preclinical data from a drug, and the pKa of that drug is 1. Bracken, are you going to let that drug go to the next level? It's a very strong acid. Do you want to put a very strong acid in your body? No. No. So your drugs will not be strong acids or strong bases. They'll damage tissue, they'll irritate tissues. So drugs will either, if they are acids or bases, will be either weak acids or weak bases. They run the heat in here, it's dry. Just also, I'm talking all the time, so the moisture goes right out of my body. So if we look at a titration curve, essentially, for an acid or a base, essentially, the percent of it that is not ionized, ionized is going to look like this. So for the acid, as the pH goes up, the acid now will start to become ionized. The base, as the pH goes up, it will start to become non-ionized. And this is the part that this is the implication essentially of the Henderson-Hasselbalch equation. This is the piece I really want you to grab hold of. The acid, as the pH goes up, will tend to be ionized. The base, as the pH goes up, will tend to be unionized. So there are some drugs that are weak acids, but more of them are weak bases. This will affect their bodily distribution. Let me give you an example. So this is aspirin. This is acetyl salicylic acid. Acetyl salicylic acid. So that carboxylic acid group on the top there is the acid part. And the pKa of that carboxylic acid group is about 4.8-ish. 4.6, 4.8, pretty much in that realm. Okay, let's just say 4.7 to put it in the average there. Now, in the stomach, that ester bond right there, should have brought my laser pointer, but I left it upstairs. That ester bond is going to get hydrolyzed because esters are acid labile. You're going to therefore produce acetic acid. And you'll then have this moiety here, which is called salicylic acid. Now, salicylic acid can ionize. It can ionize to a negatively charged salicylate and a hydrogen ion. What's the pH in the stomach? Is the stomach acidic or basic? Acidic. Insects have a basic stomach. Mammals have an acidic stomach. pH in the stomach is about 2. And if you've eaten, if you got food in it, it's going to be more like 2.4, 2.8, something like that. If you haven't eaten in a while and your stomach's starting to rumble and the parietal cells start making acid, oh, maybe it'll be 1.8, 1.5, something like that. But 2-ish is a pretty good ballpark figure for the pH of the stomach. Is that going to be, if the pKa, and again, I want you to think of this. If the pKa of that is 4.7, the pH in the stomach is 2, is the salicylate going to be ionized or non-ionized in the stomach? Predominantly. No. Go back, Bracken, go back to this. Go back to this. So the pKa is about here. It's going to be non-ionized. That's why acidic drugs tend to be absorbed in the stomach pretty efficiently. So you think of aspirin, first gets absorbed in the stomach. There's another drug, it's an anti-seizure medicine called phenytoin. That's also weak acid. That gets absorbed in the stomach, and that's why phenytoin for seizures works so fast, because you get it absorbed as soon as it hits the stomach, gets absorbed, tends to work pretty quickly. So the form that predominates, again, go ahead and if it helps you, draw this out and use colored pens or something like that. And for acid, use red. For base, use blue or something like that. And that may help you. So here are some weak acids. Here are their pKa's. Some of these are drugs we will talk about. And as you look at those, you can see some of these, therefore, are going to experience pretty good absorption in the stomach. Here are some weak bases, and here are their pKa's. And these are some drugs also that we will talk about. Now, if a drug is not charged, it can diffuse, and it's small enough, which most drugs are, they can diffuse through the lipid membrane. But they can only diffuse through the lipid membrane if they're not charged. So say you have a drug on one side of a barrier, and it goes back and forth between this charged and uncharged version. The uncharged version makes its way across the lipid membrane. If it gets charged, it can get trapped in that compartment. Otherwise, it will simply go back to the uncharged version, diffuse back to the other side, and the concentration will equilibrate essentially on either side of the barrier. But if you have pH differences such that the charged version now predominates when it gets transported, now it can get trapped in that compartment. So what are the proportions of the charged and the uncharged molecules? Depends on the pKa of the molecule, and it depends on the pH of these different compartments. And here are some pKa's, some pH, excuse me, of these various compartments. So we see urine tends to be on the acidic side because of the phosphate that's used as a buffer in our blood. Breast milk tends to be slightly more acidic than blood, slightly. Lower small intestine, because of the bicarbonate released by the pancreas, tends to be more alkaline than blood. Stomach is way acidic. Prostatic secretions, because of the citrate in it as a buffer. The citrate will actually make it slightly acidic. And then vaginal secretions from Brunner's glands and other things like that and peg cells and that sort of thing, those will tend to be more on the acidic side. Also, the presence of lactic acid bacteria usually make those secretions on the acidic side. Now ask yourself, how will this affect the distribution of a weak acid? Now, as you think about that, how will this affect the distribution of a weak base? Let me give you a case study that shows you how this can be applied clinically. Let me just read this to you. 26-year-old man is brought by his friends to the emergency department of the city hospital because he has been behaving strangely for several days. The 26-year-old man is a known user of methamphetamine. Now, methamphetamine is a weak base. It's a weak base. So stick that in your memory bank right here, weak base. He has not eaten nor slept in 48 hours. He threatened to shoot one of his friends because he believes his friend is plotting against him. Well, that doesn't sound good, does it? On admission, the man is extremely agitated. He appears to be underweight. He's unable to give a coherent history. What do you ask a patient usually to give a coherent history? Do you know where you are? Do you know how you got here? Do you know what day it is? Those are your big three to start with a history. If they can't answer those three, and say someone's had a seizure, don't remember their name. If you ask them where they are, if the ambulance brought them in from a seizure, they won't remember from when the seizure started. They'll say, well, given that you're a nurse, I'm probably in a hospital, but I have no idea how I got here. Fine. So this person cannot give coherent answers to those three questions. Not good, not good. He must be restrained to prevent him from walking from the ED and from walking from the ED into traffic on the street. So he's a threat to himself. Blood pressure is 160/100. That good? No, that's way too high. What's normal blood pressure? 120/80 is 160/100. 100, that's pretty close to hypertensive emergency. If it was over 100, now it's starting to become a hypertensive, well, crisis at least. Heart rate is 100. Temperature is 39 degrees centigrade. Respirations are 30 respirations per minute. What's normal respiration rate? Between 12 and 18 to 20, something like that. He's 30. So he's huffing and puffing. His heart's really going. So his sympathetic nervous system is jacked. The guy's jacked, essentially. His arms show numerous evidence of numerous intravenous injections. Remainder of his physical examination is unremarkable. After evaluation, he's given a sedative and fluids. And to get rid of the methamphetamine, he's given ammonium chloride. Ammonium chloride. To help him, you need to get rid of the methamphetamine. To get rid of the weak base methamphetamine, you give him ammonium chloride. So methamphetamine is pretty freely filtered by the kidney. If you administer ammonium chloride, it will take the urine and it'll acidify it. If you acidify the urine, now what happens to the base? You get a larger fraction of the drug now being protonated. That will charge it. Therefore, it will be poorly resorbed. Therefore, once the methamphetamine gets into the urine, because you've acidified it, it's trapped. Won't get reabsorbed. So you increase the rate at which you get rid of it. See how that works? Pretty cool, huh? So if I convince you this is important and you should pay attention to it, and it's not, oh, it's chemistry, I'm gonna, no. Pay attention to chemistry. Chemistry is important because it affects treatment regimens sometimes. Okay. Are there any questions about any or all of this before we move on? Yeah, Bracken.
So just looking at the biorecombinant majority of the body is on the acidic side.
Slightly, yeah, slightly. This will have implications when we talk about breastfeeding and drug therapy, because breast milk is slightly acidic. So your weak bases will have a tendency to be excreted into breast milk and then get trapped there. So weak bases will get excreted into breast milk much better than acids. Most drugs that are weak acids, you don't have to worry about, they don't get in the breast milk very much at all. Whereas the weak bases are the ones that are going to get in the breast milk and be trapped. Some drugs will get excreted into breast milk and then they go from the breast milk straight back into the blood. So the amount of drug in the breast milk is very small. So you don't have to worry about it because the amount the child will get isn't going to affect the child because the child will just get rid of it quickly. And the drug never gets to that level where it's going to cause the kid trouble. It's those weak bases or drugs that the baby cannot get rid of. Like the big one you never give to a mother, breastfeeding mother, is phenobarbital. Babies cannot excrete phenobarbital and it builds up in their body and it causes massive sedation. And so the baby will stop feeding, the baby will just want to sleep all the time, the baby will be drunk, and you never give phenobarbital to a mother because of its tendency not just to be effectively excreted into breast milk, but also the inability of the baby to get rid of it. So yeah, so the answer to your question is yes. Most of it is close to neutral, tending partially towards a more alkaline view of the world, well, slightly acidic view of the world in many cases. All right, so let's now talk about something really rather different. So how did we get to where we are in terms of drug regulation? We need to talk about the drug schedules. How did we get from the drug schedules from where we started? So you must understand that originally, not just the United States, but also Europe, experienced essentially a wild, wild west when it came to drugs. People would travel from town to town with bottles that were labeled as cure-alls and things like that. Some of them were spring water with maybe some tarragon stuffed into it or something like that. People claimed it would cure your rheumatism to whatever. And it was very much a caveat emptor, buyer beware sort of situation. You could sell anything, claim anything for it, put anything in it, and nobody would really prosecute you. This became a problem when the army started importing painkillers from the East, and the painkiller that was used was opium. Some of the opium that came into the country turned out to be adulterated. Instead of a white powder that was opium, it was a white powder that was mainly chalk. And during the Spanish-American War, when that chalk was used to relieve the pain of wounded soldiers, it didn't work. So in response to this Drug Import Act of 1948 was passed. This was the first legislation that tried to regulate drugs, and it didn't regulate drugs made in the US, it regulated drugs that were imported. So it was an import act that said if you're going to sell the United States drugs, they can't be adulterated. So this was about adulterated drugs coming in overseas, and there was an agency that was tasked with testing and determining if drugs were adulterated. The technology back then was not very good. Literally, the agents would open the container, take the powder and taste it. That was the way they tested it. It was so, but it was the first attempt, at least, to try to address this issue of imported drugs. When it came to food as well, foods normally with an agrarian society were eaten essentially straight when they were harvested, or you would take your wares to market. Most of what you ate was local. And if somebody sold garbage, you just tell your neighbor, they wouldn't buy their garbage and they'd go out of business. So essentially, there really wasn't need for a whole lot of regulation because so much of it happened at the local level and the regulation largely was through the market. When people started moving to the cities, and you started getting packaged foods that were canned, livestock that was shipped now to stockyards. The big one, of course, was Chicago. And then people started trading futures on them and stuff like that. So there was a lot of money involved with this. And when people could package foods and essentially put fillers in them, and people really didn't know what was in these, there were a lot of additives that were being used for preservation and taste. They'd never been tested. So there was a guy named Smith. There's a book that's about him I have upstairs about this guy. He recruited a bunch of bachelors who lived in a couple of tenements around Washington, D.C. to come in and have free breakfast and lunch. And he added these additives into the food, and then he recorded their health essentially for 6 months. And he had another group to which he didn't add the additives, and he compared their health essentially. And he was able to establish, at least over a course of 6 months, that these additives did not seem to affect the health of these volunteers. There were some additives, though, that did. And so Dr. Smith and his association with several politicians led essentially to this Federal Pure Food and Drug Act of 1906. And this particular act essentially specified there were certain additives you could and couldn't use. And that if you're going to use the additives in food, you had to at least test them and show that they at least didn't hurt somebody. Now, there was still a problem with narcotics. Have any of you seen the movie? It's an old movie now, Tombstone, with Kirk Russell and Val Kilmer as Doc Holliday. Val Kilmer is so good as Doc Holliday in that movie. Oh, it's a really good movie. In that movie, Kirk Russell plays Wyatt Earp, and he gets off the train. Earp had been a marshal at Dodge City, and he got out of being a sheriff because he had to shoot people and participate in gunfights, and he said, that's enough of that. So he went essentially to Tombstone because it was a boom town, and he wanted to essentially make money. And there's a scene where he meets his brothers, and he introduces them to his new wife, and his wife is talking to her sister-in-law, and she said, yeah, this heat, my headaches start coming back. And she turns to her and says, do you have any laudanum? You know what laudanum is? Tincture of opium. Tincture of opium was sold openly on the market. She became actually addicted to opium, and she died because of opium overdose. And the movie kind of documents her descent into this terrible, terrible addiction from which she suffered. But people were getting addicted, not just to things like opium, but other drugs as well. You could get them through the mail easily. You could order them through the Sears and Roebuck catalog. So the Harrison Act was meant to try and stem this easy access to narcotics. That's why it was called the Narcotic Act. Now, keep in mind, these acts came up, but the enforcement regimen to enforce them was not great. So these acts didn't have a lot of teeth. They were on the books, didn't have a lot of teeth. Well, the big act that came next was the Food, Drug, and Cosmetic Act of 1938. This act essentially created the modern FDA. And it was put into place, one, because it was clear that the 1906 Act was inadequate. Congress agreed. The bill got stalled. And then in 1937, there was the sulfanilamide disaster. This is a really sad story. So sulfanilamide was one of the first antibiotics that was brought to market. It was hailed as a wonder drug. Franklin Delano Roosevelt's son-in-law had a very, very bad streptococcal infection. It was killing him. His tonsils swelled up. He couldn't breathe. And then a doctor came and administered sulfanilamide. And within 5 days, he was better. He was out playing tennis again. And everyone wanted this drug because children were getting streptococcal infections and they were dying from it. The problem with sulfanilamide is that it's a sulfa drug. It smells and tastes horribly. The other issue is that it's not very soluble. So Massingale Corporation took different chemicals and tried to dissolve it, and they discovered that there was a solvent that would dissolve it, and that solvent was polyethylene glycol, antifreeze. It's poisonous. So they put this concoction together and then sweetened it with raspberry tasting syrup. So it was called an elixir. And they sold these containers of sulfanilamide elixir. They went all around the country. Doctors prescribed them and people started dying. And who gets sick and gets treated? Usually by doctors, children. Children started dying, hundreds of them. The FDA sprang into action, but under statutory authority, they actually couldn't seize the canisters. Then they discovered the canisters had a labeling error. Now they could seize them. The nation, as you can guess, was outraged. And this prompted the passage of this 1938 legislation. The FDA now really came out looking like the heroes in this event. They now became an agency with a bigger budget. They had a lot more agents. And this was the first law that specified, look, if you're going to make a drug, you have to prove that it's safe and not going to kill people. You have to prove it to the FDA's satisfaction. So this was the first turn on the road that led to the modern drug regulatory regimen. The Durham-Humphrey Act in '51 established a demarcation between over-the-counter and prescription medicines. So some medicines, the experience with them was so extensive, it's clear they were safe at certain dosages. Keep in mind, any drug is a poison at a certain dose. Are drugs safe? Sure, at the prescribed dose. Any drug at certain concentration is a poison. It'll kill you. So just keep that in mind. The over-the-counter medications you could get at a drugstore, at a hardware store. Aspirin was one of the biggies. There's just so much experience with it. People had experience with it. It's not an issue. And so this essentially is what's responsible for the prescription over-the-counter demarcation. The Harris-Kefauver Amendment came in the wake of the thalidomide disaster in Europe. And now the testing regimen had to be extended to human trials. And those human trials essentially were divided into the phase 1, 2, and 3 trials we talked about before. The phase 1 trials only test safety. Phase 2 trials test efficacy. And the phase 3 trials are bigger trials with people who are sick. And then this led to the Controlled Substance Act of 1970 that gave us the drug schedule regimen. So this act established the DEA, the Drug Enforcement Agency. And the drug schedules are based on the degree to which a drug will engender dependence, either psychological or physical dependence, the tendency for the drug to be abused, and whether or not, the third thing would be whether or not the drug has an established medical use. Established medical use. We are out of time, so on Monday, we'll start in with the drug schedules. We'll take a look at the difference between a schedule 1, 2, 3, 4, and 5. We'll talk about clinical trials, and then we'll get into pharmacodynamics. I looked through the pharmacodynamics section. I shortened it, and I simplified it for this year. I'm going to try it this way this time, and hopefully, I think maybe it won't be so heavy as it was before. I think you'll thank me for it, I hope. All right. Okay, so I will see you guys on Monday. Have a great weekend. Don't forget, next Friday, a week from today, we will have our first quiz. Okay?